Okay, so I'd like to welcome you all to our fourth global webinar to celebrate fungi worldwide. And of course, we all know that fungi are crucial to the functioning of Earth's ecosystems and indeed are crucial in our everyday lives. And this is something we're going to celebrate today and hear lots about many aspects from around the world. We've already had one session this morning, which were talks given from Australasia and Asia. And now we're moving to session 2, and the talks will be given by participants from Africa and from Europe. And of course, the third session, which will be this evening's session in the UK, but at different times of the day, of course, around the world, will be from speakers in the Americas. Most of the talks will be recorded, so if you miss anything, you'll be able to see it or hear it later on at your leisure. We're having 4 talks now, and each speaker will talk for about 30 minutes. And there won't be questions immediately after those talks. The questions will be in a block after all 4 talks have been given. So please do put any questions you have in the chat, and I'll read them out later on in about 2 hours' time. So actually, you can see our 4 speakers waiting to talk, and I think we'll move straight along to the first talk, which will be given by Ahmed Abdel-Azeem. And Ahmed is from the Suez Canal University in Egypt. He's professor of mycology there. And Ahmed's interests are quite varied, in fact. He's interested in ecology, taxonomy, secondary metabolites, endophytes, conservation, all sorts of things. And Abdul, if you could share your screen any moment now, that would be great, please, because Abdul is going to give the first talk, which I'm very much looking forward to, and it's called Fungi in the African Civilizations, History, Present, Conservation, and Future Prospects. So Abdul is going to tell us everything there is about fungi in African civilizations in the past and today. So over to you then, Abdul. Ahmed, sorry.
It's okay.
I've known you for a long time. I shouldn't be making that silly mistake now, should I?
No problem. Hello, everybody. It is my pleasure to be here with all of you. I think most of all of you is attend my presentation, or some of them worldwide. This presentation related to what we said, الفطريات in Arabic, champignon, Fungi and the African civilization, history, present, conservation, and the future perspective. الفطريات in Arabic, because the majority of Africa is speaking the most dominated language is Arabic, followed by French, and later on by Swahili. So I am write fungi in 3 words. And this photo, the background of my presentation, is the most important one to all of you, to see the Holy Valley in St. Catherine, where Allah talked to Prophet Moses, which give you impression that is Egypt is arid habitats. We never forget what already the ancient Roman Pliny the Elder wrote. Always something new came from Africa. So all the time we are expected some new came all the time from Africa. And we have different factors, as dear Lynne started here introducing the event, that is fungi are very important for human living, for ecosystem, for everything in that life. So you should know that total length of fungal mycelium in the top 9 centimeters of soil is more than 450 quadrillion kilometers, which equals half of width of our galaxy. So this is a huge amount of living organism under our feet, which already absorb about 70% of carbon and consider the main warriors against the climate change. So fungi protect our life. We have 54 countries in Africa, which already showed about 30 million square kilometer, and the current population is more than 1.5 billion population in all Africa. And we have different ancient Egyptian African civilization started in Egypt, Carthage in Northern Africa, and Tunisia, Morocco, Algeria, Oxium in East Africa, in Ethiopia, Numidian in Northwest, and Noba in south of Egypt, and right now it's called Sudan. So we have different kind of empires also. Ashanti, Kingdom of Congo, Empire of Mali, Kingdom of Zimbabwe, and a lot of ancient civilization in Africa, which already work directly or indirectly with our fungi. Egypt considered the cradle of mycology according to already what mentioned by Aurora in 1985, who said ancient Egyptians believed that the mushroom were plants of immortality. That has attracted me and my team many years ago, since 2000, to run in all directions in ancient papyrus and also in temples in Egypt to document anything related or similar to a mushroom all the time. And we already found a lot of interesting documents and data related to uses of mushroom and travels by ancient Egyptian. And this is already published in many papers online since 2016. And all the time, we put in our minds that ancient Egyptians are the leaders in using yeast in fermentation. So yeast is considered one of the most prominent fungi in ancient Egyptian civilization, along with mushroom, amanita, psilocybe cubensis, and other kind of mushroom looks like travels also. Already I documented this nice Hathor goddess, which already carrying amphora filled with psilocybe cubensis mushroom in her hand. And this is also documented by different clusters of mushroom all over the country, which temples cover upper parts of Egypt. Looks like Hathor temple and also Abu Simbel temple. This bag or this amphora gives the idea to the Veronika Brimova, the fashion designer from Ukraine, to make her own style of this bag filled with psilocybe cubensis. And after that, also, we found real documents for using of yeast for manufacture of wine and beer in ancient Egyptian civilization, and documented all the time in their deceptions in temples and in papyrus. And when we go to the some catalogs of Boston Museum, and which already published by Berlin in 2005 and by Abdel-Azeem et al. In up to 2017, they found that a lot of ear studs and decoration are directly related to mushroom, especially Amanita muscaria. And also Berlin, along with Abdel-Azeem, they already working to, they found already something very, very nice, which already present in British Museum related to the Cobra goddess. And I will display some nice photos for you. Here is ear stud, stone ear stud in ancient civilization, Egypt, ancient civilization, which decorated looks like Amanita muscaria. And this is already documented in what already Berlin published in his article in 2005. Here, what I'm talking about, Cobra goddess, and please concentrate your eye on this Amanita muscaria inside the cobra. I already directed or give announcement by the red arrow. Inside the cobra, you will find the bilious of Amanita muscaria and its stalk, and there is a correlation between the poisonous of this cobra and the Amanita muscaria inside it. Also, different kinds of deceptions related to mushroom present in most of Egyptian temples. Some guys is correlated between Ankh, the immortality key of life, and mushroom. And they said it's already a kind of a cross-section of Amanita muscaria, and the Egyptian artists don't distribute it or don't engrave it in a good way. And that is already, if you go through this Ankh, you will find that cross-section through the Amanita, and they correlated between Amanita and Ankh, the key of immortality in ancient Egypt, according to Berlin 2018. One nice finding is what already mentioned in Hearst Medical Papyrus in about 1550 BC, which already mentioned in his medical papyrus that the ancient Egyptian uses the moldy bread to cure their wounds. And that is give us an indication that is the ancient Egyptian using that moldy or that green fungus related to penicillin. And this is already documented in Hearst Medical Papyrus. And some nice paper is published in Mycologist correlated between molds in ancient and the more recent medicine. In 2005, Steve Berland has published his nice article related to ethnomycological origin of Egyptian crowns. Egyptian crowns is two kinds of white and red crown, and he mentioned that this idea of emerging of this crown for the king of ancient Egypt is correlated to pens of Psilocybe copiensis. So he gave us some indications that is Psilocybe copiensis during its growing gives the idea of white crown or clustered of red crowns of all ancient Egyptian kings. And some documents correlated between the spiritual uses of psilocybin in the ancient Egyptian, and you can find them already published in Scientific American many years ago and in the New Yorker journal. Through Middle Cemetery Project, which already meaning the oldest place in Egypt, in Abydos, which considers the Black House of ancient Egyptian. Myself with American team led by Professor Robert Blanchett and myself, we are working on deteriorated wood there, which already we discovered some nice catches inside the wood. These fungus dated back to 6,000 years ago or more. And here we already working to examine the deteriorated wood in Sohag digging house. And after that, we doing some isolation of the recent fungi from different localities and from deteriorated wood in ancient Egypt. And we discovered that nice catch already showed a fruit body inside a piece of wood and dated back to 6,000 years ago. Here our catch is a fruit body of a scoma of some fungi is covered under sand for 6,000 years ago, and inside the wood of coffins of some ancient Egyptian monuments. And this is also a TC cross-section in a wood from this area. And it's, you can see the mycelium of the fungus inside the wood vessels. And this is already published. You can read it in Bolos 1 as example, supplemented with all figures. Paul Stamets, already a famous guy working with psychedelic mushroom, start to come to Egypt last year and to start to document some of the ancient Egyptian psilocybe catches on different temples, and we already start to announce for Abdel-Azeem and Paul Stamets award to document psilocybe copienses or psilocybe species in all of Africa. And we start to document this even in every place we can catch in Egypt, and even in Dijot temple in Madrid, because it's Madrid has a complete pharaonic or ancient Egyptian temple there. And this is the announcement of the award. So if anyone from Africa here is already catch a psilocybe, he can go and apply for the award. It's $1,000 US. And also we are doing research, continuous research, because we are going to different circuit lakes around the temple to collect the soil and to analyze DNA correlated to psilocybe copiences in the next few months. Egyptians are leader in pusa or beer manufacture in the ancient Egypt, and they produce this drink or this beer, which contain about 2% of alcohol, and they are using it by preparing it by a very, very old simple method, by growing wheat grains and mix it with some shamsi bread or sun bread and make it to ferment many days, and after that they can got it to drink to heal their kidney disease or nurture as a nutritional food for them, because they all the time working under some hard condition for cultivation or something like that. So Buza and beer is manufactured by yeast in ancient Egypt, and this is documented also in temples. And here we are in al-Qa'ab in Abydos, and we are beering a closed vial dated back to 5,000 years ago of wine and beer manufactured by ancient Egyptians. Also, lichens go deep in the ancient Egyptian civilization. They use lichens for many targets, some of them for decoration of their jewelry boxes, and also for keeping the mummy inside the mummy against bacteria and against fungi, because some lichens already produce a lot of antimicrobial substance. Besides that, they extracted some red colors, looks like or seen from certain lichens, and they used it in painting their drawing inside temples, and this color till now is permanent, not faded through the time. Another important part for ancient Egyptian civilization is manna or travels. And Jewish know that during Exodus, when Moses took Jewish and leave Egypt, and Allah sent for them manna from the sky to start to feed it and to withstand the bad condition of the desert. And some guys from Iran told that is manna is maybe Asplisia esculata as a lichens, but indeed this lichens under surveying in Egypt and occupied Palestine, we never recorded this lichens at all. But manna, we discovered that manna is the travels, according to the hadith from Prophet Muhammad sallallahu alaihi wasallam. And we have already different types of manna or travels in Egypt. They are about 5 species, and they are already under threat due to overcollection and climate change. And they are used all the time in different applications for food, for eye drops, for give a health, vitamins for the old people. And here is in Egyptian desert, we are using our hand just to dig and to catch the travels. We are not using dogs or pigs because it's associated with herbs, not with trees. And also local Bedouins still now using seals and and bodaxes in desert for eat and for curing their injuries during walk in the desert. So here is a list of desert travel species in Arabian Peninsula and the neighbor countries, and what already some of them is all the time under threat due to climate change and overcollection. And as I mentioned to you, they are using it as an eye drop, and they got because it's treated eye diseases and a lot of problems in eye, so they are using it as eye drops. When we go towards Algeria, sorry, in the north west of Africa, we will find a Tassili mushroom cave, which already documented some uses of psychedelic mushrooms in drawing inside this cave. And also we can find something related to a man, his head and his fingers is already correlated with Amanita muscaria. So it is documented in Tassili mushroom cave in Algeria. Besides that, ethnomycological survey of traditional usage of indigenous knowledge on desert travel is very important thing, because it's Algeria is using all the time desert travel for ethnomycology and uses for treatment of people there. And this is also put a stress on desert travel. Related to ethnomycology in Eastern Africa, which related to Ethiopia and Eastern countries, Kabatsi and Mushani in 2023 doing a great article review related to that issue, and they found the majority of them are used in medicinal uses, and they are 82 genera within 39 families, and belonging to Basidiomycota, and Ascomycota just 4 genera within 2 phyla. And you can read more information about this in this nice article about how can local people domesticated the wild edible mushroom in Eastern Africa, and they produce it in a large scale production. In Africa, we have also mushroom and the travel historical biofactories, a nice article published by Hisham Al-Anshasi in 2012 with his co-worker, discuss how many travels species and how many Basidiomycota used in Africa, and in which kind of treatment. Is it as food? Is it as a kind of a treatment of sexual problems, whatever? So he mentioned in this article, how can you find this species related to the country and its uses and its application all the time. So it is one of the most important article review related to ethnomycology in Africa. In West Africa, they are using another recent research, not recent, it's published in 2014, is related to ethnomycological awareness, because people there maybe use them to in a wrong way, and the different taxa there may be over collected, and after some time it's completely threatened. And this is some common species related to macro Basidiomycota and Ascomycota in west of Africa. Fungal diversity in north of Africa is till now we have not any solid checklist related to the various countries, you know, Egypt, Libya, Sudan, Tunisia, Morocco, Algeria, and except the recent one published by Abdel-Azeem, who already mentioned 2,477 species documented in Egypt. We haven't a checklist except another one published for Libya by Abdel-Azeem al-Bouni, old one in 1980 and not updated till now. And the other countries is just checklist for Basidio, but not for the whole groups of fungi. So in north of Africa, we are met some trouble till now related to the how many species of fungi are there in north of Africa. In East of Africa, a lot of studies carried there, and they showed distribution related to fungi in different families in Tanzania and Kenya. And this is published by Tubawa et al. In 2014. But it's all sometimes, but give us an indication about the distribution of taxa among different phyla. In West of Africa, there is a nice published work by Biberbein of Germany and her co-workers related to how many species through the literature. She found 19 species in Guinea-Bissau and 1,595 species in Sierra Leone. So already documented from the literature, followed by different species, more than 16,000 records of fungi represented 4,843 species and infra-specific taxa found in many publications related to West of Africa. So this is considered a core material for us for document fungi in Africa. For Central Africa, more than 700 species collected and described. 78% is Basidiomycetes, and the rest belonging to Ascomycetes, and this is especially for Cameroon. Southern Africa, the Basidiomycetes consists of 1,008 species belonging to 251 genera and 72 families. And this is a start to both South Africa fungal database, because we have a lot of work there related to micro and macro, but the documentation to both a whole list of the country is still under work there with our team. If you go to a recent publication, which already surveyed 26 countries in Africa, they are estimated the underutilized wild edible fungi and their undervalued ecosystem services in Africa. What about the Arab countries? North of Africa, we are using just truffles for eat. We have still mycophobia. People in Egypt, people in Tunisia, in Algeria, not used to eat wild mushrooms. They are afraid to be poisonous. But in other countries, they are more than in Africa. More than using of wild mushroom in their food in comparison with North of Africa. Why are fungi in Africa vanishing? We have different habitat loss and fragmentation, prolonged drought, ultraviolet radiation, pollution, climate change, and over-harvesting, because it's many poor communities over-collect mushroom, and that is a big problem for fungi in Africa. And already we have different groups of fungi never touched in Africa. Some looks like Trichomycetes, rock-inhabiting fungi, black yeast, Myxomycetes, entomopathogenic, nematophagous fungi, mycorrhizal, endomycorrhizal fungi. They are overlooked all the time in Africa. No one interested to study them, and that is Bush, Biberbein, and the team, David Minter, Paul Kirk, to doing that mapping mycological ignores related to checklist diversity pattern of fungi known for West of Africa, and my team do that also related to Egypt. We need to know that is strategies and the level of conservation of African fungi localized in 4 types of strategies: in situ conservation, by conserving a natural habitat, and it is a big problem for us because it's climate change control everything right now. In situ conservation on mycological reserves. Indeed, mycological reserves are very, 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 very few in Africa. Even if you will find natural protected area, but not mycological reserves. In situ conservation with ecological corridors, and ex situ conservation, and we can't say it's herbarium because we changed this word completely many years ago. So related to fungal conservation in Africa, our information is data deficient. We are facing all the time data deficient. We haven't data at all related to that. And right now we said it's a fungarium, not herbarium anymore. So our Suez Canal University, I founded there Suez Canal University Fungarium, and it is already registered as the only fungarium in the African continent till now. So we have different threats, as I told you before, related to African fungi, climate change, human population growth, economic status of Africa, and over collection of macrofungi, vulnerability of African rivers to pollution, bioprospecting, and the fragility of desert ecosystem, which already causing over collection of fungi and macrofungi to extract some potential materials, and also some laws and legislation permitting often coupled with corruption. Sure, because some laws is just to protect plants and animal and overlooked fungi, lack of fund for fungal and the collection, collection, fungal project. They all the time consider the fungi under plant, and they give money for flora, not for fungi, and it is a big issue and a problem. Lacking of infrastructure, center of excellence, and training courses. And the most important thing is public awareness of fungal conservation, because it's very low in Africa till now. Here, what already published with David Minter in 2011 related to Rio Convention CBD report, which already many African countries included fungi inside plants under flora, not as a separate kingdom. Even Robert Whittaker introduced it as a separate kingdom in 1970. We did it in Egypt, and we changed that CBD report many years ago after many years of looping and stress on Egyptian Environment Affairs Agency. So the question right now, are we gonna to conserve the pathogenic fungus? Yes, you should conserve all type of fungi, because it's Rio Convention is just said, please conserve animal, plants, and not recognize bad animal or bad plants. They said conserve the living organism, and we are working to even to conserve everything related to fungi, even the pathogenic one in culture collection. So that is, you put on your mind moral issue of Rio Convention. You should keep in your mind this is a living organism. You should protect it, and you are not alone here. And we are 9 billion people, and fungi or pathogenic one are very small in comparison with us. So you should conserve all kind of fungi. Factors affecting fungal conservation of in Africa: misrepresentation, fungi or plants. We are all the time both fungi under flora. Absence of adequate fungal documentation. We haven't a recent checklist from each country related to its content and genetic resources of fungi. Limited checklist updates. Also, fungal conservation is considered a new fashion in Africa. Educated challenges to fungal conservation, and also bioprospecting versus conservation. Very important points, 6 points controlling and limiting fungal conservation in Africa. So Egypt as a case, David Minter already founded his International Society for Fungal Conservation in 2010. And in Egypt, we founded the second international or Arab Society of Fungal Conservation in the world in 2013. And we carried a lot of activities since the last 12 years to making protocol of cooperation with different associations worldwide and local, and also workshop related to fungal conservation, conference, first international conference on fungal conservation in the Middle East and the North of Africa, second international conference on mycology in MENA and on MENA and the effect of climate change of fungi in 2018. And already we founded Egypt National Fungus Day in 2016, and we considered one of the leader countries worldwide who already founded Egypt National or a National Fungus Day dedicated to the Egyptian fungi. And I think here your friend David Minter will say a small thing about that.
Actually, Ahmed, we can't hear what David's saying, so I think maybe you need to move on from there.
Yeah. Oh, okay.
And if you could start to wind up as well, that would be good, please.
Okay. So David is talk here about that Egypt is lead country and announced for National Fungus Day. And also we already founded what we said Egypt Mycologist Network. We are collected all mycologists in the country in a database, doing mycology services delivery and doing a lot of laboratory training, not in Egypt also. We founded Iraqi Mycologist Network, and we trained them for many years, and also Algeria Mycologist Network, Libyan Mycologist Network, Pakistani network. And also we doing a nice thing is related to conserve fungi through art, and invited a lady from Iran. She already doing some nice drawing and both coined them with ancient Egyptian queens, and we published them online for free or a poster card. And her name is Farouz Shafi. She's doing that for Egyptian to attract young generation to protect the fungi, and even paint myself as a guard of the Egyptian fungi. So this is a kind of interested thing to attract young generation. 3 years ago, I announced it with the help of CBD, the annual Fungus Day of Africa, which already happened annually on 25th of May each year, to help all Africa to understand the importance of mycology all over the world, all over Africa, and to support them to protect the fungi in Africa. So we have 4 challenges of fungi in Africa. Number 1, biodiversity and taxonomy and the ecology of macrofungi. We have missed data till now. Biochemistry, biotechnology, and the pharmacology of African fungi, because it's a lot of research going to over-collect these fungi and to extract of them very important substance and overlook their importance to the ecosystem. Fungi culture collection. We have only one documented in the World Directory of Culture Collection in Suez Canal University related to Fungarium, and very small 2 or 3 distributed in Africa. Also, therapeutic potential of African mushroom in human preclinical and clinical studies. It's a very important point of view, because some people transfer their fungi outside of Africa, and they dedicated them for another international institute to work on it without caring about Nagoya Protocol. So here is a summary for the challenges you already meet in Africa. misrepresentation of fungi or plants. All of people, majority of them consider fungi under flora, absence of adequate fungal documentation, limited checklist, fewer number of mycotaxonomists. The people who identify fungi are very few in Africa. We are 3 in Egypt, as example, who already using keys and identification. Also wrong and bad taxonomy. Some guys is not a mycologist and they give their expert to identify fungi. So that is why David Minter published his article, Botanist and Zoologist, Fungal Conservation Needs You. And also extension of fungal taxonomy by respecting versus conservation, neglection of systemic mycology at higher education institutes, the course of related to taxonomy of fungi, very, very neglected, and even molecular identification and the single loci phylogeny, along with low or middle income countries. Africa countries are low and low income, the majority of them. We can do any molecular studies except we travel outside Africa and doing that and pay or even a grant or a scholarship. Solution and opportunities, the end of my presentation. We should doing high performance computing facilities, make publicity available to researchers that need to work with large data set. We need technology to sequence genome from a small amount of starting material. Looks like handy nanobore. Also, conventional identification confirmed by molecular techniques. You are all the time said we need to identify by molecular. Molecular is not give you the real identification 100%. Sometimes it happen some errors. Also, we need also to work with a reduced cost of genome sequences, and this will be happened due to establishment of the African Center of Excellence in Fungal Research, which already become a leading organization in Africa, carrying out research training and community development in key areas of fungal diversity and application, in addition to integration with currently established fungal center in Africa and the world. This is a very important for all of us in Africa. And I am asking all of you from UK, from America, from Australia, Asia, to be a partner and to be a partners in this center. We need to establish it. We need to establish it in Suez Canal University. We have some infrastructure, but we need to establish the African Center of Excellence by all of your cooperation. This center will look after plant pathology and endophyte, biodiversity and chemical discovery, fungal diversity, mushroom growing, farmers and community services, microbial and microbial bioresearch center. We will support the sustainable development goals for United Nations. So we're asking you again to support the establishment of this center in Suez Canal University. Finally, please join our new AFRIMA. We already revived the African Mycological Association. So the right name now is AFRIMA. So you can join it and you can find that free of a charge to click on Google form and fill your information and be a member in African Mycological Association. It will be our happiness. Thank you for all author of all literature used in this work, especially Rosemary from Cameroon, Mohammed bin Zayed Species Conservation Fund. Everyone in my life has taught me something. The moderator of this symposium and all of you, thank you.
Thanks, Ahmed, for fascinating insights into the use and depiction of fungi in Africa thousands of years ago, before most of us had any thoughts of civilizations around the world. And thank you for pointing us to the very, very important topics which need to be acted on very, very quickly in the future. Thank you. May I now introduce our next speaker, who is Deborah Louisa Narh Mensah. And Deborah is represented sub-Saharan Africa. And she did her PhD at the University of Pretoria on biotechnology, and then more recently has worked as a senior research scientist at the Food Research Institute in Ghana. And has now, though, moved to be a postdoc at Stanford, so on the west coast of the USA in the San Francisco Bay Area. And it's a delight to invite Deborah to speak to us. And huge thanks for talking to us now, because this is very early in the morning, because as I say, Deborah has now moved to the other side of America, so it's probably about 5 or 6 o'clock in the morning now. So very great welcome and thanks, Deborah, who's going to tell us about deciphering molecular mechanisms of fungal iron homeostasis. And do try and keep it simple for all of us. Welcome, Deborah. Would you like to share your screen now?
Yes, thank you so much, Lynn. So, yeah, I'm going to try to make it as non-academic as possible. I hope you can hear me all right.
Yes, it's perfect.
Okay. So, all right. I hope you can see the screen as well. Okay, so what is fungal homeostasis? We are talking about iron is actually an indispensable molecule for several biological processes in various organisms, including human beings, animals, and plants and fungi. They are used, there are various mechanisms that organisms use for ion uptake, transport, storage, and then these mechanisms are influenced by various factors. And actually, abundance of iron causes phenton-like reactions that can cause damage to various organisms. Therefore, it's very important for these organisms to control iron balance. And that's why they have various mechanisms for iron homeostasis. And then some of these mechanisms include availability of antioxidant enzymes, free radical scavengers, and metal chelating molecules or proteins that are also known as siderophores. These compounds are low molecular weight compounds that are used for ion uptake, transport, and storage. And they are used for various purposes, such as bioremediation, growth, pathogenicity, and virulence factors in various organisms. And they serve as pathogen control substances, such as antibiotics. The biosynthesis and transport of these molecules occurs by use of genes that are located in secondary metabolite gene clusters or operons. And these follow various pathways, which I will not delve into too much. And these mechanisms are widely investigated in bacteria and some fungi belonging to the Ascomycota. So there's a huge gap concerning these mechanisms in basidiomycetes and other fungi. So now let me talk about armillaria in terms of talking about the molecular mechanisms used to, the kind of research that can be used to investigate these mechanisms. I'm going to use the example of armillaria. And these are mostly pathogens of woody plants, and they belong to the family Physalicraceae. They have a worldwide distribution. Most of them are, they range between saprophytic lifestyle and pathogenicity. Some are also mycorrhizal. Majority are known as facultative necrotrophs, which means that they can change between the pathogenic lifestyle, saprophytic lifestyle, and various forms in between. These organisms cause what is called the armillaria roots and stem rots. And this is very devastating to forestry, agriculture, and various sectors. They have a broad host range, and they survive under various environmental conditions. Some of these armillaria are also edible, and they have some medicinal properties. So at this stage, I'm going to talk about genomics and bioinformatics of secondary metabolism or ion homeostasis in malaria. These have been published in these 2 papers. So the reason for studying this is to look at the secondary metabolites, diversity and distribution of these in published genomes of malaria and other species in the Pessalacraceae, to identify siderophore biosynthesis gene clusters using comparative genomics approach, and then to putatively characterize the main biosynthetic genes in these genomes, and then to bridge some knowledge gaps generally about mechanisms of ion homeostasis in malaria. We developed this workflow for the comparative genomics and bioinformatics work. Based on this work, we identified that these, the genomes of all these studied malaria and other species in the Pessalacraceae have a broad range of putative secondary metabolite gene clusters in their genomes, ranging from 22 in Pseudobatrachydon turidi to 56 in Malaria borealis. And we also identified what is known as the non-ribosomal peptide synthase gene cluster in all the genomes. That is at least one of these. The second gene cluster that was of interest is what the software we use called Fungi identified as siderophore gene clusters. So I'm going to delve deeper into these 2 types of gene clusters because they are more linked to ion homeostasis. Now, looking at the genomes, what we wanted to do was investigate whether these gene clusters identified in all the genomes we studied were more conserved, which means that the property of producing these specific secondary metabolites are conserved across the various species in the genomes. So looking at this figure shows that each line shows the secondary metabolite gene cluster in the respective genomes. And it shows that generally, most of these genes are conserved, both in terms of the types of genes in the gene cluster and their orientation. There were few kind of mutations that we observed, such as duplication events and inversion events. This means that generally there's microsynteny and that these gene clusters are conserved within the genomes. We use a second type of protocol or workflow to determine the second gene cluster that I mentioned, which was identified as siderophore by Fungi. And we use this method to identify the gene clusters and to characterize these gene clusters. So what we detected was that there were typically 2 different types of these gene clusters in each of the genomes. Again, as we can see here, there's microsynteny that shows that the gene clusters are conserved across the various genomes. And this bracket here shows the main gene cluster, and it shows that these particular genes, this is the main biosynthetic gene, this is a transport-related gene, and this is like a protein modification gene. Now, the second gene cluster, as you can see here, consists of totally different types of gene. It still has the main biosynthetic gene clusters gene here, and then it is followed by totally different types of genes compared to the first gene cluster. So our understanding was that this particular gene cluster is totally different from the first one. And that's our hypothesis is that these 2 gene clusters will synthesize totally different types of siderophores. And looking into this particular gene cluster, the Malaria novae, there seems to be an absence of the main biosynthetic gene. By looking closely, we see a synteny between that particular region and the various, the 2 adjoining gene clusters. That shows that this may be an annotation issue, or there may be an actual mutation that prevents transcription of this gene to form the protein. Now, these various, there are different types of these main biosynthetic genes. So we wanted to see what type of gene, what type of these specific main biosynthetic genes were. There are 5 different types: types A prime, B, C, and C prime. What we detected was that the main biosynthetic gene in the first gene cluster was different from the main biosynthetic gene in the second gene cluster. Again, we showed that both of these main biosynthetic genes belong to the type A prime type of NIS synthesis gene. What this shows is that, as we predicted in the previous slide I shared, these particular genes will synthesize different types of siderophores. Now, we also conducted in vitro studies to see the actual biosynthesis of these genes, because the presence of a gene cluster or some specific gene does not necessarily translate to production of the final product or exhibition of the phenotype you expect. So we use these two in vitro assays. We use what is known as the universal chromosome S, and then the split chromosome S assay, and we compared that to the growth of the culture in growth medium. We also conducted an assay where we looked at the effect of iron on growth of each of the culture we investigated. So we used 10 strains of Armillaria species, and the medium was 1 PD potatoes, dextrose, pectose, agar, with or without iron supplementation at 100 micromolar. And then we checked for radial growth and then macromorphology of these cultures. We also used what is called the liquid chromosome S assay to determine the effect of iron concentration on siderophore biosynthesis of 2 strains of Armillaria species. Now, what we detected with the solid media chromosome S assay was that these are the controls showing the universal chromosome S, the split chromosome S, and the actual growth medium. We detected siderophore production showed by the color change from blue to various colors on here. Now, we also detected that depending on the assay used, whether it's universal chromosome S or the split chromosome S, we detected different types of siderophores. We imagined that because siderophores are produced using amino acids as the substrates for the main biosynthetic gene, we imagined that the difference in siderophore biosynthesis is based on the difference in amino acids present in the respective media. But then this also tells us that it is important to use different media when you're trying to determine siderophore biosynthesis by various organisms. Now, this same image also shows that siderophore biosynthesis is actually strain specific. Looking at Armillaria fuscipes, we use 2 different strains here, and we see some differences in siderophore biosynthesis in terms of the quantity of siderophore produced and the intensity. The intensity shows the affinity of the siderophore produced for iron. So the stronger the color intensity, the stronger the affinity of the siderophore produced for iron. So it shows that these organisms are strain specific with respect to iron homeostasis. Now, looking at the assay we did with the 2 different iron iron in the media, we also observed the same kind of trend where the effect of iron on the growth of the culture was dependent on the strain. For instance, looking at Armillaria gallica, strain CMW31092 had a significantly increased culture growth rate compared to the one without iron supplementation, while the reverse was observed for the second strain. So again, the relationship of each strain with iron is strain dependent. So it's very important to assay various strains of the same organism to actually have a real picture of what's that organism or how that organism interacts with whatever compound you're looking at. Again, we observed some brownish exudates in the media. This was either diffused in the medium or appearing as liquid on the culture. And we observed that this was generally more prominent with iron supplementation. We expect that these exudates are actually secondary or other metabolites and would require further investigation to see the composition or the actual metabolites being produced. Looking at the general culture morphology, generally the culture morphology was maintained with or without iron supplementation. But then we observed that the culture colony morphology of Armillaria napsonina strain CMW3159 showed more dense culture morphology with iron supplementation, while CMW4456 showed some apical hyphae, whitish apical hyphae with iron supplementation. Again, this goes to show that the kind of effects that the molecule iron has on the cultures are strain dependent. Now, looking at the fact that these organisms were having different relationship with iron, we wanted to see at which points that siderophores would not be produced by two cultures. So we looked at supplementation of iron at different concentrations in the liquid medium, up to supplementation up to 200 micromolar of iron. Now, what we observed was that the trend was different as compared to what is usually seen in the literature, where there is usually an inverse correlation between iron supplementation and siderophore biosynthesis. This shows that Armillaria has a specific, unique requirement for iron, and this needs to be investigated further. Now, most of the work that I have just talked to you about was generally concerning genomes that had already been sequenced. So we said we should sequence a genome of Armillaria from Africa, and we did that with the culture from Zimbabwe. This has been published. And then we used that genome to do comparative proteomics investigations this species in response to iron. And basically, we wanted to assemble and annotate a genome from Africa, an Armillaria genome from Africa, and to make it publicly available for various research. We also wanted to gain further into the insights into the types and extent of iron-responsive proteomics and secretomics that occurs within the specific isolate, and to unveil some of the iron-dependent mechanisms employed by Armillaria species. So we use general standard DNA extraction and genome sequencing protocols. We use both long and short reads. We assembled these reads, and then we conducted genome assembly, quality analysis, and various standard protocols for genome assembly and annotation. Now, what we observed is that the genome we sequenced had properties that, or parameters that are comparable to genomes of other Armillaria species that have been published in the literature. And this particular genome was, it had a translation and location factor 1 alpha that was identical to the one that has been deposited in GenBank. So this is generally a good quality draft genome sequence of this particular strain of Armillaria species that is now publicly available for further research. Now, we use standard protocols for protein extraction, trypsin bead digestion, and then peptide concentration. We use gel-free and label-free LC-MS to sequence the proteins, and then we use bioinformatic tools to analyze the sequences that were generated. Now, based on this research, we used 3 different biological replicates of each of these treatments that we did. So we used this PCA analysis to see the repeatability of the biological replicates. This shows that the 3 different replicates are generally, comparable in terms of the proteins identified. And it shows good repeatability of the experiments we conducted. It also shows that there's a ion-dependent effect of effect on the protein that is secreted or produced by mycelia in the secretome, as in the growth medium, and also in the mycelia of the strain we used. These are the control, that is the proteins that were in the growth medium. These were removed from the experiment for the actual cultures, and that shows that this reflects the actual production of proteins by the actual mycelia and secretome. Now, this image shows that the more proteins were significantly increased in abundance with ion supplementation in the mycelium. That is the proteome in itself. Then more proteins were decreased in abundance in the secretome. And then there were also a whole lot of proteins that did not show any significant change in abundance. Usually, most of these proteins in this range are ignored, but I'll tell you why sometimes it's important to look at some of these proteins as well. So generally, we looked at the gene ontology, annotation, and functional classification of the proteins detected in the proteins. This is actually to tell you what types of proteins are being are having a significant change in abundance, and then to link that to the actual function of these proteins. This gives an insight into what kind of biological processes are being altered based on the condition you provided. So in this case, with or without ion supplementation, what we realize is that the carboxylic acid cycle is favored in terms of central metabolism. More enzymes in the TCA cycle were significantly regulated or increased in abundance with ion supplementation compared with no ion supplementation. And this goes to show it's extra proof that ion plays a significant role in the TCA cycle. There were a lot of amino acids that were increased in abundance with regards to amino acid biosynthesis and secondary metabolism. We also observed that a lot of proteins related to growth were increased in abundance. Now, this is very telling of the fact that we saw about a 50% increase in culture growth with ion supplementation of this particular strain. So looking at the proteins that showed no significant change in proteins, in protein abundance, we saw that most of these proteins are linked to oxidative stress or metal toxicity. This shows that if these proteins were increased in abundance, you would understand that this particular supplementation, 100 micromolar, is toxic to the organism. But once these proteins are not significantly up-expressed or up-regulated, it tells that this particular ion supplementation is not toxic to the organism. So the organism did not need to increase the abundance of these specific proteins that are generally increased in abundance when the organism is experiencing ion stress or some metal toxicity. So the general conclusions is that we have now one good quality publicly available genome of an Armillaria species from Africa. We have the first record of siderophore biosynthesis by Armillaria species. We have the first evidence of the ion bioaccumulation capacity of Armillaria species. This includes the first records of a highly conserved, potentially functional, non-ribosomal peptide synthase gene cluster, 2 distinct NRPS independent siderophore synthase gene cluster in fungal genomes. Then the gene cluster is typically able to synthesize and release different types of siderophores at different rates. We also have the first proteome and secretome of an isolate of Armillaria from Africa. And we have identified potential targets for inhibition or attenuation of pathogenicity or virulence of strain CMW4456. What are the future perspectives of this research? We found that although we found some good answers to some of the questions we raised, there are still a lot more gaps in terms of genomics, biological and biochemical mechanisms of secondary metabolism and ion homeostasis in the Bastidiobiota. We need to conduct studies to fully characterize the genes in the predicted secondary metabolite gene clusters, studies and applications of siderophores from Bastidiobiota in agriculture, forestry, bioremediation, and medicine, and to use a multidisciplinary approach, including biology, biochemistry, pharmacology, and other techniques to understand these mechanisms. And then generally, although this particular research is linked to Armillaria, this can be extended to various Bastidiobiota. And this actually highlights some of the issues that Ahmed mentioned in his presentation. Now, some of the future perspectives, some of the possible benefits of suggested research, the research that I just suggested, and the application of the new technologies include research on all the research gaps I identified. These will contribute to attaining 4 of the Sustainable Development Goals of the UN, which is reduction of poverty, quality education, and decent work and economic growth. This includes postgraduate research training, postdoctoral and other research opportunities, job in the pharmaceutical and other industries. The knowledge and technologies for limiting these plant pathogens will contribute to some of the other SDGs, and then the application of siderophores produced by Armillaria and other organisms and their analogs can be useful in agriculture, forestry, bioremediation, and medicine. And this will also contribute to, for instance, zero hunger, good health, and life on land, pertaining to the Sustainable Development Goals. I would like to acknowledge all my funding bodies, the University of Pretoria, where I conducted this work, specifically in the Forestry and Agricultural Biotechnology Institute, the NRF for funding my PhD research, the CSIR Food Research Institute, where I was a staff at the time. And I would like to acknowledge my advisors at the time, Prof. Martin Coetzee and Prof. Brenda Winkville, who were phenomenal advisors. I'd like to thank you all for listening. Thank you all for time, and I'll be looking forward to your questions. Thank you.
Thank you, Deborah, for telling us about how we can use genetics, comparative genomics, and proteomics for understanding how fungi maintain iron balance. That's given us a good insight into some of the complex research being conducted in Africa. Thank you so much for coming along, especially as it's so early in the day for you. So now may I welcome our next speaker, who is Jacob Heilmann-Clausen. Jacob works as a professor at the University of Copenhagen in Denmark, and Jacob is interested in biodiversity and conservation of wood decay fungi in particular. And Jacob's going to tell us about combining citizen science, traditional taxonomy, and barcoding to expand knowledge on fungal biodiversity in Denmark. And of course, we all know that for a very long time, before the word citizen science was even coined, mycologists contributed a huge amount of data, which we've in more recent times been able to analyse and answer questions about how climate change, for example, is affecting fungal diversity and activity. But Jacob has really done a great job in mobilizing citizen science and doing it in a very, very scientific way. And I look forward very much to hearing what you've got to say. Welcome, Jacob.
Thank you so much, Lynn. And welcome. Well, hello to everybody. It's nicer to see people in reality, and I can just add that we yesterday had, this weekend have a fungal festival in Copenhagen, also at the university. So there we had the possibility to talk with many citizen scientists and students and ordinary citizens interested in the mycological world. So it was just fantastic. But it's also amazing to be able to share some of my work with you. And I guess you do see my screen now, is it correct?
Yes, it's fine, yes.
Super. And you see it in the presenter mode? No, you don't.
No.
Now it should be better, right?
Perfect.
Super. So I will talk about how we have worked with citizen science in the Danish Fungal Atlas and how we are now extending that work into a European project. So let's get started just with giving a little, this is mainly a repetition of what Lynn just said, that citizen science is really something with an emerging potential. When you see these records here, these graphs are showing how many records of different organism groups that are shared in GBIF, the Global Biodiversity Information Facility. And you can see the fungi are the dark green here. So we are way beyond groups like insects, plants and birds especially. But it's increasing to some extent, but still, I mean, there's a lot of unknown diversity out there, and there's a lot of potential to add more data to the global platform. Also hearing these talks from Africa, I mean, there's a huge potential to, I mean, it's the good thing about knowing not too much is that there's a huge potential to increase knowledge, and I really think that's important to bring on the table. But oops. Yes, and as Lynn also said, it's not new that kind of amateurs, non-professionals are contributing to fungal science, to mycology. So here's just two examples that are, at least in Denmark and Europe, pretty famous. Jacob Elaine, who made a 5-volume book on the Danish agarics, Flora Agaricina danica, and F.H. Møller, who made the first monograph of what is now known as agaricus in Europe and Denmark. And both are kind of classical works on agarics in Europe, and both of these persons were teachers that did this as a extra job. So it's not new; it's an old tradition, but now it's coined as citizen science, and that's what we are now working with in the Danish Fungal Atlas, which started out with the first funding from the Ove Jensen Naturfond in 2009 and run for 5 years, initiated actually by a citizen scientist also, at least a non-biologist, Jan Westerholt, who passed away in 2011. He was a geographer, so he was at least academic, but he fell in love with fungi and started this fungal atlas and got the funding to start it up. So in this first period, we had actually good funding with 2 full-time positions for mycologists to help people identify the fungi, to provide identification keys, and to organize field camps to increase awareness and knowledge about fungi, and also to go to parts of Denmark that were less known and involve all the fungal amateurs. Then we got a second period of funding that is actually running out this year after some extensions. It was not planned to be more than 5 years, but because we had some big jobs, especially in relating to this big book about basidiomycetes that we just published last month, well, 2 months ago, we were able to get funding for updating our databases, to provide more outreach in terms of publishing books, both on, I mean, the main results, what we learned from the first Atlas period, but also this book with identification keys that I'll come back to for Danish basidiomycetes. We also got funding to make a mobile app for recording in the field with a mobile phone, and I'll come back to that later also. And I mean, by combining these different efforts, we have actually put Denmark pretty high on the fungal biodiversity map. So here you see the split-gill Schizophilum communis, that is one of the few globally distributed species, at least in the temperate to tropical zones. And you can see when you zoom in, the single hexagon with the highest number of records is actually Greater Copenhagen. So we really made it into the map of fungal records. Of course, there's many records out there that are not shared yet, but at least for those records that are shared globally via GBIF, we are really well ahead in Denmark. And we kind of introduced many. We were very lucky in this second funding period to have a programmer that was also a fungal amateur, Thomas Stjernegaard Jeppesen, who is now working for the GBIF, and he really made a really fancy setup for us, including a really sophisticated way of validating records, so that we have a very high standard for the quality of the identification work that we have, so that it's a little bit like, if you know iNaturalist, you can have different identifications for the same record, and then all users in the system can vote for these IDs and also suggest a new alternative ID if they believe they have a better idea of what is already present. So you can have basically infinite number, but normally less than 5 different species IDs to each record. And then we have 2 ways of validating records. We have expert validation by more or less professional mycologists, and then we have most of the work is done actually interactively with everybody able to contribute with their votes. So everybody can vote for the record, and then it's adding, you can see the number down here for this Cortinarius obtusus was 74, and now it's expert validated as a different species. But these weights are calculated automatically based on the rarity of each taxon in the database, the geographical distribution within Denmark, and the phenology. That means what time of the year the fruit bodies are formed. And then also each user has a weight in the system depending on the proven experience in the particular group of fungi, and also with each single species. So it's a pretty sophisticated setup that is working completely automatic, and is meaning that if I, as a non-lichenologist, is finding a lichen, it will have a very low likelihood to be proven correct, but if I find a garrick, it will be very different because I have a very good rating within these groups. So it's working very seamless to validate our records, and basically 3/4 of our records are simply validated by this kind of interactive community validation, and then another 4 are expert validated, and then some records end up being non-validated or only recorded at the species level. Then 2 years later, 5 years ago, we were also able to launch our own AI system to identify fungi based on the photos that people submitted. So it was basically run by some computer scientists from the Czech Republic that were winning a competition where our data set with photos from the Danish Fungal Atlas was the training. So they made the best model to identify the fungal species based on all these photos, and then we collaborated with them to implement that model into our mobile app and also to our web system. So we now have a system that can identify more than 1,500 species. It's also using habitat and substrate data to provide reliable identifications. And I mean, when we started this up, I was very skeptical. I never believed that, I mean, fungal fruit bodies are so kind of morphologically unstable in the presence. They can look very different in dry and wet weather and develop a lot over time. So I thought it would not work very well, but actually we are up to having with this kind of human validation in the loop, more than between 80 and 90% of the identifications that are suggested by the AI are actually proven to be correct as far as we can judge. So I think it's really a good tool for our fungal amateurs. And even for me, when I find fungi that I can't remember or I have never seen before. So it works amazingly good. And all this has led to increase of users over time, especially the launch of our mobile app with the artificial intelligence, which was also highlighted in the Danish television at the primetime news, brought a lot of attention, of course. So we run down from being around 200 users every year to now being more than 1,600 users in Denmark submitting records into our database. And that, of course, also means that our number of records has gone up from around 60,000 each year to now around 70,000. So it's not a huge increase. So it's also showing that building these very kind of user-friendly tools are maybe not so good for attracting not needed to attract the really interested that are already eager to participate, but rather is a way to reach out to the greater public domain to show that, oh, here's a tool that you can use. And maybe some of these people that are in the beginning not very active are ending up being very active contributors because they realize that this is fun and doable. You can actually identify fungi and help us making this data available. So we can see that the number of records per user has dropped. So we have a lot of these casual users that are just submitting a few records, and it's not uncomplicated to have such a change in the community because, I mean, back in what we call Fungal Atlas 1, we basically knew all our contributors. That's not the case anymore. So there's a lot of people that we need to help, and we don't always have these resources. So it's sometimes a struggle to have many users in such a system. So we really gone from this dedicated community to an open platform with many casual users that we have to interact with for good and for bad. Our data has, it's called citizen science. That's of course about making science, but it's also relevant to add the term conservation or nature management because our data is also used for that. So first of all, we have some high nature value maps for Denmark where records of red-listed fungi are used to highlight natural areas with high conservation value. And it's even been used in some of the work we have done on spatial prioritization for nature based on complementarity, where not only birds and orchids and butterflies and beetles are contributing to modeling which parts of the country needs to have special emphasis if we want to conserve all the biodiversity of Denmark. Fungi are actually counting in with 1/4 of the species that are analyzed for that. So that was one of the first important aspects of the first funding, that we should make data on fungi available for nature conservation, and we actually succeeded pretty well in that. So that's nice to see that fungi are now not a neglected group in nature conservation in Denmark, but is actually placed pretty central, well, at least to a limit, well, to a reasonable extent, is because there's a lot of fungi we don't know about, of course, but those that we know about are actually considered equally to plants and birds and so on when nature conservation is done in Denmark. We have also produced research on data on many different aspects, from bias in how citizen science data is collected across different habitats and in urban versus rural areas. On collaboration with, among others, Lynne Boddy, we have also shared data across different countries in Europe to work on fungal macroecology and diversity patterns across Europe, and we also did a paper on that actually brought us to the front page of New Phytologist on host selection in Danish wood inhabiting fungi. So we also could make a tick mark on the science part of this data, even though there's a lot of issues with noise, and it's not like standardized data that you collect as a professional, but the strength is that you get a lot of data, and a lot of data you would never be able to get if you only worked as a professional. Then the last thing we started to add some 5 years ago was to sequence, focusing on barcode regions, the ITS region, to help us make this new book that is out in Danish with the keys to more than 4,600 species of basidiomycetes. We hope it'll be out in English next year, so it'll be available for everybody in Europe and throughout the world that are working with fungi that are present in Denmark also. So it was a big job to do that. It's still working with spores and everything, but to build the keys, we sequenced almost 5,000 collections of fungi, mainly provided by our citizen scientists. And we had a very high success rate of getting good reads out of 97%. Could add almost 400 new species to Denmark based on those 2,000 species we got sequenced in total, but also finding a lot of species that are not really matched in gene bank. That might be new species, but in reality, it's often species that are just not barcoded yet. And some of these are actually in the process of being described. So even in your own backyard, you can more or less go out and find new agarics, even in a country like Denmark that is highly populated, highly researched for fungi, and with very little nature remaining. So there's work to be done. You don't have to go to tropical rainforest or the deep sea to find new species if you're a fungal amateur in Denmark. And just some examples of how it has expanded our view, for instance, hypnums, which are known edible fungi that are very popular among fungal foragers. Until 20 years ago, we believed that there were 3 species in Denmark, but now we know that there's at least 11, probably even more. It's possible to identify most of those based on a combination of macroscopical and microscopical characters, and they're all edible, so it's not a major problem. But in terms of finding out how these species select their habitats and so on, it's important to know that they are different biological species. And of course, it's even worse in some of the big groups like Cortinarius, where we now are up to more than 450 species in Denmark. We sequenced 331 different species, and of these, almost more than 40% were new to Denmark, and increasing the number of known species a lot. And we also found species that are completely new to science. So just to take one example, Cortinarius inulius, which is a well-known group in species in Europe. Well, group is the right name because we realized that we at least have 8 different species. Most are probably very difficult to identify based on just morphology, but if we're adding in also the ecology, it might be possible to do it. And at least one species, this Cortinarius nudipes, is very characteristic with the white patches of universal whale in the cat margin. So that's just an example of where you open up the box and realize that it's much more complicated than we thought. And of course, it's even worse in some groups like these small, nice club fungi, where we sequenced 150 collections and found around 50 species, but only around 15 with a good name match in gene bank and Unite and all the other databases. So this is definitely a group where a lot of taxonomic work is needed to understand what is going on in terms of species diversity, and also in terms of ecology. These species are very poorly known, so we basically don't know what they live from. They're probably plant associated, but nobody really knows. So it's just saying that even in Europe, mycology is still a very young science in terms of finding out what all this fungal diversity is about and how it's contributing to ecosystem functioning, and how we can also learn to maybe use them in providing a sustainable future for the humanity. Yeah, this is just one example from my local forest in a suburban forest in the Copenhagen area, where we're now describing 3 different species, including 2 Cortinarius species and one Inusibe. So it's just a funny slide to show so that we're in the local forest where I walk my dog every morning, I can go out and find unknown diversity. I think that's pretty fascinating also for fungal amateurs to be able to contribute to that. So we also tried to find out, as also Ahmed said about Africa, how many species of fungi do we actually have in Denmark? And we are basically much more, I definitely think that most countries in Africa has much higher fungal diversity, but our numbers are actually higher. You can see here the curve, the species accumulation curve is actually showing exponential growth for what we know about Denmark. If basidiomycetes in Denmark, we're now up to around 3,700 species. So the blue dots here are from just before we did the DNA sequencing, and then you can see then how we continued up to the red dot by adding DNA sequencing. So this is just the increase in known species diversity over time. And the good question is, when will this curve start to flatten out? How many basidiomycetes do we actually have? I mean, we included 4,600 species in the keys because we know that there's a lot to discover yet, but I think the real number is still much higher. We know that some groups of very inconspicuous ectomycorrhizal fungi like Tomentella and Sebacina has a lot of diversity. For instance, when we look at meta-barcoding results from soil DNA, we can see that there's probably hundreds of these species remaining to be discovered and described. They're probably impossible to identify by morphology, but still they are distinct species with different ecologies and different roles in the ecosystems. And then when we then add all the fungi, it's even, of course, worse. So here's one study we did with meta-barcoding for 130 plots in Denmark, where we selected soil samples and extracted the DNA. And you can see the full data set is showing all groups of fungi, and the accumulation curve is ending up around 10,000 species, but it's not flattening out yet at all. So my best guess would be that we have between 30 and 50,000 species of fungi in total. So, I mean, there's no end to this within my lifetime in terms of just discovering fungal diversity, even in a very investigated country like Denmark. To expand a little bit beyond Denmark, we just started this Fungi project that is working to bring fungal diversity on the European map, where fungi have been neglected quite considerably in conservation and monitoring work. So we are both working with citizen science and meta-barcoding and other approaches to find ways that we can start really bring fungi into the map. So in a nutshell, it's about, we have 2 different lines of research in terms of recording and monitoring fungi. So one is to empower citizen scientists, and the other is to take soil samples and find out how we, or environmental samples, I would say more broadly, to investigate fungal communities. With the citizen science, we are working to update our picture recognition apps, and we are making specific missions where we or projects where we focus on specific groups to involve people. And then we have for the eDNA, we share a common project on pine forest across Europe to record fungi there. And one of the issues with the results you get from meta-barcoding is that you often don't get exact species names, but just these OTUs, which stands for operational taxonomic units, that you may know the genus, you might know sometimes the species, but often there's uncertainty because the traditions are not really matching. So when you want to compare what a citizen scientist can record with what you get out of a soil sample, it's not always so that it's very often so that the matches, the puzzle pieces are not matching because it's different traditions. So to improve that, we have also a huge effort to go into mycological collections and barcode the old type specimens, put them into the UNITE system so there's a stable identity to the species, so we can start improving the match. And then in the end, we are wanting to use all this data to investigate biodiversity patterns and also to evaluate how well globally threatened fungi are actually conserved in Europe based on things like the Natura 2000 and other cross-national conservation strategies. So we are then designing some citizen science project with different purposes. Currently, we have 6 different projects running. There's one that is not on our homepage, but basically everybody can apply, can participate, at least in Europe. If there's a national, there's some rules about the Nagoya Protocol. That means it's that we need to have some national collecting points responsible for doing the work, but then we can get information materials. So we have this Gastrum and some other missions to work for getting data into the international red listing of gastroid fungi that are often considered in risk of extinction because of their soil requirements and other habitat requirements. And then it can be recorded via the Pluto F-Go or app or national. We're also using the Danish fungal atlas because it's adding its data into GBIF on a weekly basis. And then we can provide maps and also link the records. So over some months, we will get the records that people send to us as dried specimen sequence, and we will provide the sequences on as a link from our homepage. I can tell more about that, but there will be a different talk in the Lynne Boddy's Friday sessions later on in October. So most of the work we're doing with sequencing is that people send specimens to us. This is from North America, where this involvement of citizen scientists is very much more developed. So there's big campaigns to sequence a lot of fungi across North America, but we also, inspired by the North Americans, want to bring the technologies out to the people by having workshops where people can actually do the work themselves with these new portable workstations for field sequencing that can really be a game changer for working with DNA sequencing in mycology. And I mean, finally, to round off, I mean, there's big global perspectives for working with citizen science. It's of course a good way to bridge the science society barrier and making science more relevant for people and also better understandable. But it's also a very good way to connect mycologists, both amateurs and professionals, and potentially across the globe. I mean, if you have a good internet connections, you can basically be part of a global community of people interested in fungi, and I think that's a really amazing potential. And then of course it's there's many ways this can be done. This is just examples of things that could be done and that we have experience with Denmark. We recently wrote this paper on what citizen science can actually do in terms of fungal conservation, that you can read more about what we think are potentials in that aspect. I mean, the biggest challenge is that it's not, I mean, even though you can do a lot, it is without resources for really bringing the things together, it's not that funny. You need somebody that can help you to reach a higher level and provide the sequencing potentials or the funding to do it. But basically, it's straightforward to involve in citizen science, and we already have the different platforms like iNaturalist and the BMS database is of course only running in Britain. The Danish fungal atlas is actually available across the globe and can be used across the globe. We just don't have the taxa from over the world, and we don't have the resources to really support it. But especially iNaturalist and this fungal, there's also this American Mushroom Observer. They are freely available across the globe, and also the Pluto F-Go app can be used across the globe. And I even think that UNITE community in Estonia is providing resources for sequencing if you can handle the Nagoya Protocol resources for sequencing. So that was basically, just to show the map again and showing also, I mean, one thing is to be proud that Denmark is so much on the map, but having the African perspective, I think there's a huge work to be done to really put fungal diversity on the map in a more kind of reasonable manner. And I really hope that citizen science can be one tool for doing that. So that was my final slide. So thanks to all the fungal amateurs, to the team of the Danish Fungal Atlas, and lastly to the Fungi team. And thank you all for listening.
Thanks, Jakob. That was truly inspirational. I think it gives us great hope that these apps for identifying, for those of us who are really rather poorer identifiers than we probably should be. And I'm sure that we're all desperate for Denmark's Bacilli of Svampe book to be translated into English. So I'm sure there'll be lots more questions soon about that. So thank you so much. And now we need to move on to our final speaker of this session, which is Elaine Bignell. Elaine is Professor of Medical Mycology and the co-director for the MRC Centre for Medical Mycology at the University of Exeter in the UK. Elaine is the current president of the British Mycological Society, and her research, as you will guess, involves human pathogenic fungi. So on that note, straight over to you, Elaine, and welcome.
Well, thank you very much, Lynn. Thank you for organising. It's been a real pleasure to be here today to listen to the incredible talks, but it's also an honour to contribute. So thanks again to you. So this talk was originally written for the British Science Festival, which was held at the University of Exeter last year. And I particularly like the mission statement of the Society, which refers to celebrating the people, the stories and the ideas at the heart of science. Because, of course, every scientific story is intertwined with a myriad of other stories, all of which provide the impetus for positive change, for the advancement of technology, and also for the freedom to live good lives based on good decisions. And it's therefore vital that we share those stories. So thank you very much for the opportunity to use this platform to highlight some of the challenges we face in medical mycology. So this particular story begins in London in 1970. So there was a heat wave. This was a sweltering summer. And the big news was that Concorde had made its first flight. The Beatles had disbanded. Jimi Hendrix had passed away. Pelé had led the Brazilian football team to victory in the World Cup. And in a suburb of West London, my mum and dad were juggling sleepless nights with raising a family. And while I was bouncing happily through the first year of my life, it was an exciting time for the scientific field as well. So the structure of DNA had been solved 17 years previously. And in 1970, my future PhD supervisor, Professor Herb Arst, here he is, pictured here, was a research fellow in the Department of Genetics at the University of Cambridge. And he was working at the forefront of genetics research. There were no genome sequences or genetic tools back then. And his toolkit included an easy to work on fungus called Aspergillus nigelans. And this was an important vehicle for genetic studies at the time. And an ultraviolet light source that he used to introduce mutations into the DNA of the fungus so that he could study the consequences of those genetic changes. It was an exciting time for medicine as well. In the United States, Edward Donald Thomas was developing bone marrow transplantation to cure leukemia. And before long, that technology had become adopted in Europe too. It was pioneered at the Hammersmith Hospital in London. But unfortunately, this positive trajectory would not be without challenges because profound suppression of the human immune system was necessary to prevent rejection of the bone marrow grafts. And this made patients highly prone to infection. where still, just a few years later, the US Centers for Disease Control documented 5 previously healthy young men in Los Angeles as having confirmed pneumocystis pneumonia, also known as PCP. It's a disease that was caused by a fungus then known as Pneumocystis carinii that had infected the human respiratory tract. And when it quickly became clear that the disease, the underlying predisposing disease in these individuals was acquired immunodeficiency syndrome, AIDS. And it quickly became clear that this disease was spreading globally and caused by a virus that has led to the death of more than 35 million people in the past 4 decades. So effectively, in just a few short years, the population of individuals who were at risk of fungal diseases, either through the advances in medical practices or through HIV infection, had expanded greatly. And as well as pneumocystis, there were several other clinically important fungi that were causing life-threatening illnesses. And I'm showing what we call in medical mycology the big 3. These are images captured via electron microscopy of Aspergillus fumigatus, Candida albicans, and Cryptococcus neoformans. These are fungi that are found all around the globe. They have significant prevalence as human pathogens, and they can all exist as microscopic, single-celled life forms as spores or yeasts, and exhibit a property that is very important for pathogens of humans. They're able to grow at human body temperature. So they are particularly thermotolerant fungi. So at the time, the world wasn't well prepared for fungal diseases occurring on such a scale. Antifungal diagnostic tools were highly limited, and there were only 3 classes of antifungal drug available. The azoles and the polyenes, which target the lipid membrane of fungal cells, and pyrimidine analogues that mess with RNA and DNA synthesis. And it would be 3 decades more until the medicine cabinet was well stocked. In the meantime, the field of medical mycology was beginning to become well established. The study of clinical fungal diseases and the pathogens that cause them. And Herb Ast, my PhD supervisor, had been recruited from Cambridge to head up the Department of Infectious Diseases at the Hammersmith Hospital. So while Donald Thomas was preparing to receive his Nobel Prize, I took up a position in Herb's lab, where I stayed for 9 years, initially as a research technician, and then as a PhD student. So at this time, it was the beginning of the end for apartheid in South Africa. The Queen has described 1992 as being the annus horribilis. It was the dissolution of the marriage of Charles and Diana, and Windsor Castle had been razed to the ground by an enormous fire. But in Hammersmith, Herb had become interested in studying how fungi were able to adapt to pH. And pH is a chemical way of describing the concentration of hydrogen ions in a substrate. And Herb had become interested in understanding how fungi were able to adapt to the pH of the environment in which they were grown. So in his model system, he'd seen that Aspergillus nigelans was able to comfortably adapt to life across a different range of pH values, even broader than the range that I'm showing you here. But for my PhD, I'd study the way in which this easy to work with fungus could adapt to high pH, and pH is 7.2 to 7.4, which are commonly encountered by fungi which inhabit the human body. So ordinarily, this fungus is quite resilient to pH changes, and that's what you can see in this top line here. So basically, each of these images has been captured from a different growth medium where we've played with the concentration of hydrogen ions in the substrate. But we found that this ability to adapt to high pH could be prevented if you exposed the fungus to ultraviolet light and selected for certain mutations that prevented pH adaptation. And this very simple experiment told us a few very important things. Number one, that the fungus is able to extract and interpret information about its external environment. Number two, that process is important for normal growth. Number three, that the mutation of DNA can prevent the ability to adapt to high pH. So this adaptation process was likely to involve genetically encoded components. And number four, which would be very important down the line, the inhibition of this process could prevent the growth of the fungus at the range of pHs that are commonly encountered in mammalian tissues. So using the tools of classical genetics, we were able to map the cause of this phenotype to a region on the far right extremities of one of the Aspergillus nigrans chromosomes. There were two genes. In fact, one was called, in the end, we called PAL-H, and one we called PAL-I. And this was the very first scientific paper that I published. So over the years, we've learned through our work, but the work of many, many, many other laboratories around the world, that this pH signaling mechanism is highly conserved in fungi. And it's incredibly important for fungal physiology because the fungus has to produce enzymes and proteins that are able to function beyond the confines of the fungal cell. So the fungus needs to be able to produce enzymes that can help it to acquire critical metal ions, as we've heard earlier in Deborah's talk, to be able to break down complex food substrates and then reabsorb them. And all of this is tailored to the pH at which the fungus is growing. And it's metabolically, physiologically impossible for the fungus to produce all of these hundreds of different enzymatic activities all at once. So the ability to sense and adapt to pH enables an economy of physiological resource. Also, although highly conserved in fungi, this is a process and a mechanism that we don't find in humans. So we realised fairly early on that potentially, if it were important in pathogenicity, that maybe this would be a druggable pathway. And as we learned more about the pathway, we discovered that the two genes that we'd identified back in the early 90s actually encoded two proteins that are present in the plasma membrane of the fungal cell. They're integral membrane proteins. And PAL-H on the left-hand side here is a 7-transmembrane domain protein, a type of protein that is incredibly druggable. And most of the drugs that are FDA approved at the current time target this type of receptor. So the fungus uses the pH sensor to sense external pH, and then the receptor signals to some other proteins downstream, which chop up a regulator protein called PAXi, enabling PAXi to enter into the nucleus of the fungal cell, where it switches on genes that encode functions required at alkaline pH, and it switches off the expression of gene products that are required at acidic pH. So down the line, the activity of the pathway became easily measurable to us by just looking at whether or not Paxi was being chopped up. And that will become relevant later on. So whilst I was studying Aspergillus nigelans in a department of infectious diseases when fungal infections were becoming more prevalent and were very, very challenging to treat, it occurred to me that I could transport the knowledge that I'd acquired during my PhD and apply that to the study of a human pathogen. And the organisms that I studied immediately, but actually now for the past 25 years, really focused on in incredible depth is Aspergillus fumigatus. And Aspergillus fumigatus is a soil-dwelling mold. It sporulates abundantly and it grows rapidly at 37 degrees. And we're looking here at a colony of Aspergillus fumigatus. This one is growing on a Petri dish. It's been incubated for 2 days from a more or less a single spore inoculum. You can see that 2 days at 37 degrees generates a colony that you can see with the naked eye. I'm showing on the left-hand side a lesion in the human lung that has been caused by inhalation of Aspergillus fumigatus spores. And the fungus here has basically degraded the lung tissue and used it as a nutrient source. At the level of histology, what you would see in the airway is the fungus colonizing the major airway. But what you can see here is that these hyphae can invade the surrounding tissue to acquire nutrients. And in the laboratory, we study this process in lots of different ways. But what we're looking at here is a time-lapse microscopy video of a Aspergillus fumigatus strain that has been genetically engineered to express a green fluorescent protein. And so what you can see here is the fungal spore germinating and then growing as a branched mycelium. And this hyperbranching morphology is very characteristic and used for diagnostic purposes still. So we now know that aspergillosis is a disease that can affect a wide range of individuals. So these diseases are rare compared to bacterial diseases, but sometimes serious if not diagnosed and treated rapidly. And it's important that we continue to develop diagnostics and new drugs. And I'm going to tell you about why this is important in the following few slides. But in a nutshell, any type of lung deficiency, be it cystic fibrosis, severe asthma, chronic obstructive pulmonary disorder, pneumonias of the severity that land you in hospital, severe influenza or COVID-19, or severe immunocompromise. These can wipe out your innate immune system and or local immunity in tissues and make you susceptible to fungal colonization. Now, the good news is that there are very good drugs to tackle these diseases, and we've relied mainly on the azoles, and these are orally bioavailable drugs, so you don't have to be in hospital to have an azole, whereas most other antifungals have to be administered through the intravenous route. The azoles are used as frontline therapy for all of these different types of aspergilloses. And in the first part of this century, there were several new classes of azole drugs that came to the market, as well as a brand new class of drugs called the echinocandins, the first of which, caspofungin, became available at the turn of the century. A problem that we're facing, though, at the moment is that azole resistance is increasing through time and space. So on the left-hand side, I'm showing you some work from Matt Fisher and colleagues at Imperial College in London. The dual use of azoles as medical and agricultural fungicides has accelerated the emergence of clinical azole resistance. So some types of azole fungicide, which are, they're used in enormous amounts in agriculture, are also able to confer cross-resistance to our first-line medical azoles. And because the degradation half-life of azole fungicides is long, this has led to significant exposure in the natural environment. We also know that infections in humans that are caused by azole-resistant Aspergillus isolates can be associated with heightened rates of fatality in hospital. So this is an important problem that requires our attention and likely in the future some altered public policy in order to contain. So the genetic signatures that drive these different types of azole resistance has greatly facilitated ongoing hospital and environmental surveillance studies that are seeking to decipher the rates and the geographical distributions of medically and environmentally acquired resistance. And this is work that is ongoing in real time. I'm showing you the data of a citizen science project led by Jenny Shelton that was published in 2022 that looked at azole cross-resistance of around 5,000 isolates that were collected by people around the UK. And in this study, 14% of isolates collected in the UK exhibited a cross-resistance to medically relevant azoles. So what do we do if we lose the utility of an important class of anti-Aspergillus drugs? Well, we have other drug classes that we can perhaps turn to, but this is not without problems. So again, we're looking at some microscopy, real-time time-lapse videos that look at the real-time responses of Aspergillus fumigatus to the other two major classes of drugs, echinocandins and polyenes. And on this side, we're looking at the fungus growing without the drug. And then these movies have been synchronized to show you what happens if you add caspofungin. So what you'll notice here is that caspofungin, although you're seeing the loss of fluorescence here that indicates the hyphae have been killed, you'll see them disappearing, the hyphae are able to regenerate. So what's not happening here is that the fungus is being killed and immediately eliminated. The fungus is able to fight back. And actually the echinocandins are not useful for treating aspergilloses as monotherapy. For that reason, they have what we call a mostly fungostatic mode of action at the level of killing Aspergillus hyphae. And this drug here, ambisome, what you'll notice about this drug is that when the drug is added, the fungus immediately stops growing. So here's the drug added now, stops growing, and those hyphae start to vacuolate and disappear. You notice the loss of fluorescence, and they don't come back. But ambisome is a drug that is associated with significant toxicity. And so it has to be used very carefully, and there are limitations around that that we need to take care of. So it's going to be important going forwards that we're able to find new ways of using the drugs that we have. Antifungal combination therapies are one way forwards. But also we need new classes of antifungal drugs, and that's work that's ongoing in the field. But it's complicated because fungi and humans, we're quite closely related, more closely than you think. If you look at the relatedness of proteins encoded by the human and fungal genomes, there are significant similarities, and those explain the toxicity of antifungal drugs that we have, and radically limit our ability to identify new classes of antifungal drug that can be used in the clinic. And most drug discovery efforts fall down at the level of toxicity studies. It's not so hard to find drugs that will kill fungi. It's very hard to find drugs that will kill fungi but not harm the human patient. So on to 2015. Well, what can I say of significance in this year? Well, Mark Zuckerberg had just found out that the number of users of Facebook had advanced beyond 1 billion. 1 in 7 human beings was using Facebook. I'd moved to the Manchester University, where I was fortunate enough to work alongside colleagues who co-established with myself the Manchester Fungal Infection Group. It was a really wonderful experience to work with these brilliant people in Manchester who are still there. I welcomed my son, who we named Donald. It was unfortunate that Donald Trump was in his first presidency. That coincided. But I thought at the time that this would be a transient problem, most likely. Time will tell. And also I became a professor in that year. I was still working on pH signalling in Aspergillus fumigatus, and the work had advanced a fair bit, actually. So we'd been able to show in Aspergillus fumigatus, by looking at that regulator protein, PAC-C, and measuring whether or not PAC-C was present and being chopped up. This piece of data just shows it's a way of imaging the protein. We could see that there were long and short forms of the protein, both detectable at low pH, pH 5, but only the short form at pH 8. So this is how we visualize the activity of the pathway using an experimental process called Western blotting. If we delete the PAC-C gene, we do that genetically using genetic engineering, then obviously we can't detect the protein anymore, and that's why these tracks are blank. But here, if we delete the pH sensor, HALH, that gene I discovered when I was working on my PhD with herb, well, then what happens is that PAC-C stays in its long form, it's not chopped up, and then the fungus is unable to grow at high pH. So what you'll notice here is that this is even more challenging for the fungus when we add sodium ions to the medium, because sodium is toxic to fungi at high pH. So if the fungus can't adapt to high pH, it becomes much more susceptible to certain ions, but also to certain antifungal drugs. Moving to mammalian lung tissues, we discovered that if you delete PAC-C, the gene that encodes the protein PAC-C, if you delete that gene, actually the fungus is unable anymore to invade the human airway or the mammalian airway. So the fungus can still generate germlings and hyphae, but they're unable to punch through that respiratory epithelium. So we'd proven that the pH signaling pathway was absolutely essential for pathogenicity. And that was very exciting because it told us that most likely we could try to target pH signaling. as a means of identifying new antifungal drugs. And so the way that we approached this was to go back, all the way back to the classical genetic work that had been done in the 70s, the 80s, and the 90s of the previous century. And we re-engineered the pH signaling pathway. We went back to Aspergillus nigelans to do this because it's such a great model organism. And we decided that what we'd do is we'd find a gene in the fungus that was regulated by PAXI, and that was going to be important for the fungus to grow. And the gene that we picked was a gene called GABA. And basically it allows the fungus to utilize an essential nitrogen source. We genetically engineered the fungus to put this gene under PAXI regulatory control. And normally PAXI switches GABA off, okay? And what that means is that in the absence of a preferred nutrient source, the fungus can't grow. And in our engineered strain, effectively what happens is that if the fungus can't sense pH or transmit the pH signal, then the fungus is unable to use if the pH signal is compromised or the signaling event is compromised, the fungus will be able to grow because the expression of the GABA gene will be enabled. So it's the reverse of a normal drug discovery screen where you might be looking for a drug that would switch off the growth of the fungus. We engineered the screen so that if we found a drug that hit the pH signaling pathway, the fungus would grow. So effectively, we were using chemicals instead of genetics to prevent pH signaling. Of course, we didn't know whether or not this was going to work, but as it happened, it worked very, very well. So this is just a set of data from a genetic screen where we used a library of 500 different chemicals to see whether or not any of them would hit the fungal pH signaling pathway. We see what we would expect is that if the fungus is functioning properly and able to sense and signal pH, the fungus won't grow. So that's this line here in the red. But if we find a drug that hits the pathway or we knock out a gene that's important for signaling, like PAL-H, then the fungus will grow. And these red dots here are examples of chemicals that we found in the screen that specifically hit the fungal pH signaling pathway. So that work has evolved into a collaboration with industry. And we've worked for a long time now with Dave Cook, formerly of Blueberry Therapeutics, and Ed Griffin from Medchemica on a unique series of chemicals, a novel class of chemicals that can specifically inhibit pH signaling. And it's very exciting to be able to develop these agents as options for inhibiting pH signaling. They may be important clinically, and that, of course, is the goal of our research at the moment, but they may also be useful for use as agents that can be used as fungicides in other domains. But of course, what we want to do is find out where their use is going to be best applied and make sure that they're developed for that purpose and hopefully for that purpose alone. Now, these days, if we think about the burden of human fungal diseases, it's not reducing. And if we think about how far we've come compared to those early days in the late 90s, the late 80s, the early 90s, when medical mycology became an expanding field of research, there are still many problems that need to be fixed in this field. So in sub-Saharan Africa, where the burden of HIV remains very high, less than 15% of African countries have access to life-saving diagnostics for common AIDS-defining respiratory illnesses. Different countries and continents are affected by different pathogens. Some of them are newly emerging. I give examples of two. Candida auris down here on the left-hand side. This is a newly emerged multi-drug resistant Candida species, which is rapidly spreading around the globe. Lots of medical mycology labs have switched their focus to the study of this organism to try to understand why it's multi-drug resistant and whether we can identify combination therapies which will be useful for treating it. Sporotrichosis, an important fungal zoonosis, which is now endemic in Brazil, is a disease that's spread by cats, fatal in cats, disfiguring in human beings, and quite often requires plastic surgery in order to correct. This disease is what we call endemic to particular regions of the world, but there was a case reported in the UK a couple of years ago, and this was via the import of a cat from Brazil. It was quickly detected and didn't become a problem in the UK, but that's not to say that it wouldn't in the future. And of course, I've mentioned the importance of thermotolerance in fungal pathogens of humans, and we have to be able to cope with the changes that global warming will elicit in the epidemiology of diseases caused by fungi in humans. So you can't go back and change the beginning, but you can start where you are and change the ending. Now, relative to other types of infectious diseases, those that are caused by fungi have long been overlooked. And as a result of that, we lack the critical capacity that we need to make the types of rapid advances that we need to address these problems. And my work at the University of Exeter, at the MRC Centre for Medical Mycology, addresses directly this problem. But I just want to highlight that in the first pre-pandemic decades of this century, the investment from G20 funders into infectious diseases research topped $105 billion US. And this funding was secured from hundreds of philanthropic, public and other funders. And it went into the study of more than 30 individual pathogens, but less than 1% of that funding can be traced back to any type of research into diseases caused by fungi. So at our centre in Exeter, we have four overarching goals, one of which is to engage funders and the public and researchers. And that's, of course, what I'm doing today through this talk. But we also want to drive ambitious advances in the field. We desperately need to build capacity. So medical mycology is a discipline that takes, a long time to become proficient with the handling and manipulation of pathogenic fungi and to study the interaction of those organisms with their hosts. So along with our centre director, Gordon Brown, and my fellow co-directors, Delia Warris and Bill Horsnell, our work at the centre is focused on research, building capacity globally, training, and also engagement. So these goals were helped recently by the publication, the first World Health Organization fungal priority pathogen list. And the World Health Organization identified a number of fungi that it categorized as critical, high, and medium priorities for increased research. A major driver in the process of prioritizing these organisms was the prevalence of antifungal drug resistance, and the fungi that are grouped in this category are Aspergillus fumigatus, the organism that I've worked on, Cryptococcus neoformans. This is an encapsulated, a polysaccharide encapsulated yeast, Candida auris, that multi-drug resistant yeast, and Candida albicans. So just a few weeks ago, we were out in Brazil to launch our CMM, Centre for Medical Mycology, Latin America unit, which is based in Sao Paulo. And this is a large part of the work that we're undertaking to nurture a global network for tackling fungal diseases. So the centre in Exeter now has 2 international centres, one in Cape Town in South Africa, and our newly launched centre in Latin America. And we're working with multiple international agencies to leverage funding to support research in these centres. So here we are visiting a manufacturing facility, but also the headquarters of Fiocruz, which is an important part of the Ministry of Health in Brazil, who are interested in supporting this goal. And that's it from me. A few acknowledgements on this slide that didn't appear on the others, but with that, I'm going to sign off and hand back to Lynne so that she can chair the Q&A.
Thanks, Elaine. That was absolutely fascinating and indeed hopeful, with the ability perhaps to target pH sensing, etc., etc. May I welcome the other speakers back onto the screen, please, so we can have some questions. But as we got Elaine there, perhaps we could start with a question for you, Elaine. Oh, incidentally, people, please do add questions to the chat. Azoles are clearly really important as antifungal treatments, but there's this major problem with using them in the environment. Do you think there's a case for banning the use of azoles in the natural environment, or is that horse bolted?
Yeah, I think, Lynne, when it comes to figuring out whether we protect our food or human health, there are enormous weighty arguments that can be put forth on both sides. Has the horse bolted for azoles? Not necessarily. I think that it's absolutely incontrovertible evidence now that the cross resistance that is acquired in the field is being seen in isolates that are entering into the clinic. So the genome sequencing studies have delivered definitive evidence of that. But there are big questions that remain about precisely, where in the food chain, at what stage and in which parts of the world is this a major problem? Is it associated with particular manufacturing processes, like composting, for example? And how can we get a better handle on why this is happening to try and limit its occurrence going forwards? Of course, the difficulty with spore-forming molds is that once they're there. And, part of the reason that fumigatus is a major pathogen is because it produces spores at such a prolific rate. The key will be in perhaps combination antifungal therapy, definitely the development of more classes of azole drugs, other classes, non-azole drugs, the ways that we use the drugs that we already have, but also going forwards, a dialogue between fungicide and clinical pharmaceutical development that somehow delivers a level of control around the way agents are used in both of those sectors.
Yeah, makes sense. Makes sense. Yeah. Actually, while we've got you there, Elaine, a question's just come up, which relates to something you were just saying quite near the end of your talk. And it asks, how does the percentage funding for viral, bacterial, fungal, and parasitic research compare to the percentage mortality and or morbidity for each of those groups?
That's an excellent question. So the study that I cited measured this relationship as well. For most of the diseases, there was let's say there was a weak correlation between the burden of disease and the amount of funding. Fungal diseases were the only significant outlier. Yes, it's certainly the case that even if you factor in the relative prevalence of those diseases, fungal diseases have been massively underfunded for the past several decades.
And the same for fungal research in general.
Yes.
Yeah, thanks for that, Elaine. Actually, so while we've just been talking about control of organisms in the field, perhaps I could ask Deborah, or do you think, Deborah, that the subject that you were investigating, iron balance, can really be used for controlling pathogenic fungi, such as when you were talking about Armillaria, the tree pathogen, but indeed perhaps other fungal pathogens of plants as well? Is there good hope for that, or?
Yeah, I think there's hope for that, because looking at the specific secondary metabolite gene clusters that I found, these secondary metabolite gene clusters are kind of unique in terms of the composition of the genes and also the main biosynthetic gene. So it's likely that the actual secondary metabolites being produced by these fungi are unique. And I know siderophores have been used as antifungal drug control substances as well. So I think that because these specific siderophores that will be produced by these fungi or are being produced by these fungi are unique, they may escape some of the defense mechanisms of these other fungi that they may be used on. So definitely we need to do the research, but I think there's hope in that line.
Yeah, thank you. So a very general question, which was aimed at Jakob, but indeed other people might want to throw in an answer as well, is effectively, when is a species in fungi? How do we know when we found a new species of fungus? What are the formal requirements? So, for example, percentage differences in the genome or different phenotypic features?
Yeah, I can try an answer. I think there's no like formal requirements, but there's different traditions. And I mean, until DNA barcoding became kind of a household business, I mean, it was mainly differences in morphology or ecology or perhaps distribution. In some cases, even crossing experiments were used, but I mean, considering how easy many fungi are to grow, it's kind of remarkable that rather few studies have actually deployed kind of spore crossing experiments. It has been done, but it's not a standard at all. So I mean, in this molecular age, it's mainly a question of really investigating branching patterns in phylogenetic trees, and especially with some of these high throughput sequencing techniques, you can actually also see the different haplotypes in one specimen pretty easy. And then it's if you have distinct clustering in the way that the DNA marker regions are organized in a tree, and you have a good sampling, it's normally the way to accept that these are different lineages, because the likelihood of genetic exchange is extremely low when you have these patterns. But it's not always solid, and I mean the same applies. I work a lot with bird people, and there's often realized that what we consider different birds, they can actually interbreed if they were forced to do it. So there's also some behavior going on in ecology and distribution patterns. But normally nowadays it's based on tree typologies based on DNA, and that's the main way to do it.
Yes, I would agree. And of course, you need to look at more than one gene as well, not just a single gene phylogeny really nowadays.
Yes. And I mean, with new technologies arising, it will be more and more full genome patterns, or at least genome skinning patterns. But still, it's very often done on few marker regions. Sometimes even only one marker region, especially if there's also support from ecology or morphology.
Indeed. So there's another question: how well does your app pick up novel taxa?
It does not. That's the thing about AI. It's only able to identify, well, it's a little bit more technical than that, but it can basically only identify species that are actually in the training data set. And that means that they have to be represented by a certain number of photos. In the newest model, it's actually able to say, oh, this is most likely not a fungus that I know, and maybe even saying this is not a fungus. I mean, you can take a picture of your dog and then you will get or your hand and you will get a suggestion for a fungal ID. So it's not really suitable for finding new species. There we still have to rely on traditional overview and of course the sequencing.
Yes, and of course, though, I suppose if a taxon came up which wasn't being voted for by many people as being, I don't know, species A, B, or C, or whatever, maybe experts would investigate that further. I don't know whether you have the resources and time to do that sort of thing, or whether that is
well, yeah, but that's more or less what we have done. But we also have been targeting specific groups where we know that the taxonomy was improving a lot and where there's very good reference databases. So, for instance, in Cotinairs, which is these small brown fungi, there's actually been done a very big job to barcode as many types as possible. So there, the reference library is nice in contrast to these club fungi where it's very terrible still.
Yes, absolutely. So just going back to Ahmed's presentation, Ahmed, you mentioned conserving fungi, and I think you mentioned through fungaria and cultures and things. What do you think is the prime way of conservation? Do you think that the types of approaches that Jakob has mentioned would be valuable for using in Africa?
The problem, okay, Lynn and Jakob, the main problem is to insert in the mind of people that is fungi are completely different from flora. So it is a big issue for us in Africa. They are all of, even in my university, they teaching in plant in systematic course, fungi under the plant kingdom. Oh my God. So it's a big issue. And fungarium, to change the mind, it's just a word, to change the mind of them. Also, the major problem for fungal conservation, not even in Africa, even in Europe, you know already what have been done by Dave, and he already run after many Darwin initiatives to set down that idea. We need a solid structure related to citizen science along with fungal conservation, because it's citizen science already doing a great work. They introduced the new taxa even in Holland, in Denmark, but they doing that related to a project, related to biodiversity project. But the concept of fungal conservation is still not inserted in the people. Even during my last visit to Romania and North Macedonia, very few countries all over the world have red listing of fungi, very few number, and the great effort of Greg Moller along with Andres Dahleberg. So the people all over the world not recognize that the most important organism in the biotic component of the ecosystem is fungi. So they don't recognize that. So through citizen science, through education, through funding, through cooperation, to say fungus is still the orphans of rain. They are still the orphans. We need to shed the light on that.
I think you're absolutely right about people still thinking about flora and fauna despite the IUCN, the International Union for Conservation of Nature, taking on board and having the slogan, the 3 Fs, fauna, flora, and fungi.
And fungi, yeah.
Is still not being taken on really by very many countries. And it is something we really need to push for. I know we need to push for that here in Britain, and certainly in very, very many countries. We need to get that into people's heads. You're absolutely right. People really need to understand how important fungi are.
Yes.
So,
Sure.
so actually, that leads me to another question to Jakob. You mentioned that I think that you got your app onto primetime TV. How did you manage that, or did I mishear what you said?
Yes, when we launched the mobile app with picture recognition, the university sent out a press release, and it was taken up by one of the big two channels in Denmark. So and it was a really interesting experience, because I was they actually put quite some effort. It was, of course, we also launched in the right season, so I could stand out in the local park close to campus and explain how it worked as the expert, and telling that, oh, of course you cannot trust this for your edible fungi. You have to really look carefully into a book, or ask somebody that really knows. So don't eat fungi just because the app says that it's edible. And then at the same time, they had another team filming with one of these kind of what is it called? And then it's a nature. Well, somebody out with a lot of citizens looking at fungi and telling about how to recognize edible fungi. And he tasted on a fungus, a russula. And in russula you can taste if a species is edible or not. So he said, yeah, in this case you can actually ask taste if its taste is good, it's edible. And then they cross-clipped that. So in the end, I was out eating that night, so I didn't hear it before I came back. Then they said, okay, yeah, so don't trust. I was saying, you cannot trust this AI for eating fungi, but then it and then they clip to this, and then if it tastes nice, then you can eat it. So so that was kind of a nightmare. So we had to go out the next day, and they had to say, oh, we really made a mistake here. And it was the social media was just crying. Somebody even called made a report to the police that this TV station was putting people in risk. So that was an interesting experience. But I mean, there's a lot of, at least in Europe, but I think also in North America, and maybe expanding to other parts of the world, there's an increasing interest in fungi, both for the good and for the bad. I mean, The Last of Us, the TV series, and the computer game, of course, but also this hope that fungi can help us make a more sustainable world by upgrading waste to food, by providing new drugs and new useful chemicals. I think there's a lot of emphasis. So at least over the last 20 years, I've seen a lot of hope for fungi in the broader society. And actually, it's sometimes the academic institutions that are leaving behind. I mean, even at the University of Copenhagen, the fungal collections are still filed under the plants. I mean, I've said that to the leader of the collections many times, that, oh, you're living in the 60s. We are now in the 21st century, but it's still the case. And there's a lot of conservatism, and even within the big funding part, I mean, there's still this tradition, and also in the primary teaching of students. I mean, you get all the cases in all the different disciplines. It's animals and it's plants. When it's population biology, when it's genetics, when it's breeding, when it's things like that. It's very rare to have fungal examples into the primary curriculum, even at universities, even in Denmark. And I think that's something that we have to work on to make a change.
Yeah, I do agree entirely with that. Yeah. Perhaps the last question then, there's one in the chat for you again, Jakob, and it's to do with the apps. And I think one thing actually I would like to add to that is, how widely can your app be used in general terms? But the particular question says, which app for fungal identification when there's no phone signal?
That's a good question. At least I think in most of these apps, I think also iNaturalist, you can use them in an offline mode. So you basically just take your photos, and then when you're back with good connection, you can then work with your records again and involve the AI. At least that's how our app works. So you have a notebook function where you can use it without having connection, just making your pictures and adding them to a GPS location that your phone will record. And then when you have the good connection, then you can start playing with the AI model. And to the specific question about our app, I think it's pretty usable in Denmark and surrounding countries. I think it's to some extent even usable in central Sweden or southern Europe, but I wouldn't use it in Africa. There you would definitely get the wrong answers because it's trained on Danish fungi.
Absolutely, absolutely. But of course, similar things could be done for training on fungi for other regions.
I think iNaturalist has a decent app, and these are developing very quickly now. There's a lot of development, and the critical mass, the critical need is a lot of photos of fungi with the right identification. Then you can, it's just a matter of running these machine learning algorithms.
Yeah. Well, thank you very much for that. It's now 3:45, and I think probably we should draw a close at this point because it's only just over an hour until we've got session number 3 coming along. So I would like to thank all of the speakers for giving fascinating presentations, and I mustn't forget to thank Sally in the BMS office for doing such a great job as well. And I welcome you back for the 3rd session, which includes talks on pathogens of invertebrates, historic uses of fungi, which is a sort of in some ways a follow-on from Ahmed's talk, I guess. That's this is from our from our American contingent. And there's a diversity in deep sea fungi and mycorrhizal biogeography. So that's on a worldwide scale so a host of different talks still to come in another well, in about just over an hour's time. So I look forward to seeing more of you then. And thank you once again. Enjoy the rest of your day. Bye-bye.
Thanks.
Thank
Thanks,
you.
Lynne. Thanks,
Lynne,
Lynne.
for all the
Bye.
Yeah, bye-bye.